You and I both work with rodents in our laboratories. How do you talk about your research with patients to, I guess, try to help them understand the importance of the research that you're doing?
Yeah, I mean, I start off with really honest discussions with people, both about the opportunities that come along with animal research and the limitations. And so the question is, what can we learn from, let's say, mice that may be inbred that ultimately applies to humans? And I think that's a really important question, right? There are lots of things that we've learned throughout the neuroscience space, whether it's in animals like squid or in mice or even in flies, about how those brain cells, those 100 billion brain cells that create electricity, how they create electricity, right? We've learned things like that they use ions, right? So things like potassium and sodium and chloride ions, and that is part of how those cells create the electrical pulses. Well, that is what we say is conserved, or some of those properties are very similar in a fly, in a mouse, in a worm, and in a human. And so that allows us to both understand how the biology is working, but also how you can target maybe how the electricity or the salt ions are moving back and forth across the surface. And it turns out some of that gives us an understanding of the things that we use in the clinic. Originally things that we use for anesthesia, but ultimately things that can be used to treat pain and psychiatric disorders as well. So I think there's a lot to learn in the animal models that apply to humans.